Hey everybody, Ronnie here. So, I did a thing, and it's the same thing I did the last 5 weeks. So, I wrote a book. It's called How Evolution Works by Ronnie Joubert. You can find this on Amazon in paperback or Kindle format. It is also available on Audible and also the Apple Store, whatever it is. I'm a Android guy myself, so but it's a decent book, I think. I want to go ahead and say, right off the bat, thank you so much to everyone that has liked these videos. Thank you for subscribing. And thank you for buying the book. Okay, every single one of you. I appreciate it. Today we are going to cover the end of part one, which part one was, the beginnings of life. And today is the rise of DNA. So far, RNA has pretty much ruled the world. Okay, we've kind of touched a little bit. This is going to really cover all of the the subject of why DNA is such a great genetic carrier. So, I hope you enjoy this. Once again, if you enjoy this content, give it a thumbs up. Don't forget to hit the subscribe button. If you have any questions, throw them in the comments down below. I'd love to answer any questions that you have. But once again, I did try really hard to make this as comprehensive as possible. So, enjoy the rise of DNA.
In the ancient waters of a primordial Earth where life was just beginning to find its footing, a momentous transition was underway. The RNA world, characterized by ribozymes and RNA molecules that both stored genetic information and catalyzed biochemical reactions, was giving way to a new era. This era would be defined by a molecule of greater stability and complexity, DNA. The dawn of a DNA-based world was marked by a series of evolutionary innovations that would forever alter the trajectory of life on our planet. In a shallow, sunlit pool rich with organic molecules and bathed in the energy of hydrothermal vents, a population of protocells thrived. These protocells, with their dynamic lipid bilayers and bustling metabolic networks, were driven by RNA. Ribozymes catalyzed essential reactions, and RNA strands served as the blueprints for the synthesis of peptides and proteins. However, the inherent instability of RNA posed a significant challenge. The single-stranded nature of RNA made it susceptible to hydrolytic degradation and chemical modifications, which threatened the integrity of genetic information. One particular protocell, nestled in a niche where temperature and mineral content created an ideal environment, harbored an RNA molecule that exhibited a novel capability. This RNA molecule, through a series of random mutations and natural selection, had evolved the ability to catalyze the synthesis of a new type of nucleic acid, deoxyribonucleic acid, or DNA. The enzyme like RNA, known as a ribozyme, facilitated the assembly of deoxyribonucleotides into a double-stranded helix, a structure that would prove to be far more stable than its RNA counterpart. The transition from RNA to DNA did not occur overnight. It was a gradual process, driven by the selective pressures of an ever-changing environment and the relentless march of evolution. The initial DNA strands synthesized by the ribozyme were short and rudimentary, serving as experimental templates rather than fully functional genomes. Yet these early DNA molecules possessed a crucial advantage. Their double-stranded structure provided a protective mechanism against the harsh conditions of the primordial world. As the ribozyme continued to refine its DNA synthesizing capabilities, the protocell began to accumulate more substantial DNA fragments. These fragments, stabilized by hydrogen bonds between complementary bases, proved to be far more resistant to hydrolytic cleavage and other forms of chemical damage than RNA. The double-stranded DNA also allowed for the development of new mechanisms for genetic repair and replication. When damage occurred to one strand, the complementary strand could serve as a template for accurate repair, ensuring the fidelity of genetic information. The enhanced stability of DNA provided a significant evolutionary advantage, allowing protocells to maintain larger and more complex genomes. This, in turn, facilitated the evolution of more sophisticated regulatory mechanisms and metabolic networks. The additional genetic material encoded for new proteins, some of which played roles in the replication and repair of DNA itself. The emergence of DNA polymerases, enzymes capable of synthesizing new DNA strands with high accuracy, was a pivotal moment in this evolutionary journey. These polymerases replaced the ribozymes, catalyzing the formation of phosphodiester bonds between deoxyribonucleotides and ensuring the faithful replication of genetic information. As DNA became the primary genetic material, the protocell's internal processes underwent significant changes. The transition to a DNA-based world necessitated the evolution of a new set of molecular machinery. The ribozymes that once dominated the protocell's interior were gradually supplanted by protein enzymes, which offered greater catalytic efficiency and specificity. These enzymes, encoded by the DNA, facilitated a wide range of biochemical reactions, from DNA replication and repair to protein synthesis and metabolic regulation. The central dogma of molecular biology, DNA to RNA to protein, began to take shape during this period. The DNA within the protocell's nucleus-like region served as the repository of genetic information. Transcription of DNA to RNA, mediated by RNA polymerase enzymes, produced messenger RNA or mRNA, molecules that carried genetic instructions to the ribosomes. These ribosomes, themselves complex assemblies of RNA and protein, translated the mRNA sequences into proteins, the workhorses of the cell. The evolution of the ribosome was another critical development in the transition to a DNA-based world. Ribosomes, with their ability to accurately translate genetic information into functional proteins, represented a significant advancement over the simpler ribozymes of the RNA world. The emergence of transfer RNA or tRNA molecules, which matched amino acids to the corresponding codons on the mRNA, ensured the correct assembly of polypeptides. This translation machinery enabled the synthesis of a vast array of proteins, each with a specific role in the cell's metabolism, structure, and regulation. The increased stability and complexity of DNA allowed for the development of more intricate regulatory networks. Genes could be turned on or off in response to environmental cues, allowing the protocell to adapt to changing conditions. Regulatory proteins, such as transcription factors, bound to specific DNA sequences and controlled the transcription of genes, ensuring that proteins were produced only when needed. This level of control and coordination was essential for the efficient functioning of the protocell's metabolic and replicative processes. The transition to a DNA-based world also had profound implications for the evolution of cellular structures. The increased genetic complexity allowed for the differentiation of cellular functions and the development of specialized compartments within the protocell. Membrane-bound organelles, such as the nucleus, endoplasmic reticulum, and Golgi apparatus, began to take shape, each performing distinct roles in the cell's overall function. The segregation of genetic material within a nucleus provided an additional layer of protection and regulation, ensuring the integrity and proper expression of the cell's genome. As the protocell evolved into a more complex and organized entity, it became capable of more sophisticated interactions with its environment. The development of sensory and signaling pathways allowed the cell to detect and respond to external stimuli, such as changes in nutrient availability or the presence of other organisms. These interactions were mediated by a variety of receptor proteins and signaling molecules, which transmitted information from the cell surface to its interior, triggering appropriate responses. The rise of DNA as the primary genetic material marked a turning point in the history of life, setting the stage for the diversification and complexity that characterize modern organisms. The enhanced stability and capacity for information storage provided by DNA enabled the evolution of larger genomes and more intricate regulatory networks. The transition from RNA-based catalysis to protein-based enzymatic functions increased the efficiency and specificity of biochemical reactions, supporting the development of complex metabolic pathways and cellular structures. The dawn of a DNA-based world also laid the groundwork for the emergence of multicellular life. The ability to store and regulate vast amounts of genetic information allowed for the differentiation of cell types and the formation of tissues and organs. Multicellularity provided numerous advantages, including increased size, specialization of functions, and the ability to occupy diverse ecological niches. The cooperative interactions between cells within a multicellular organism required sophisticated communication and coordination, facilitated by the intricate genetic and regulatory networks that evolved during the transition to a DNA-based world. As we continue to explore the evolution of life, the rise of DNA as the primary genetic material offers a profound example of how molecular innovations can drive biological complexity and diversity. The story of the transition from RNA to DNA underscores the dynamic and adaptive nature of living systems, highlighting the intricate interplay between genetics, metabolism, and cellular organization. By understanding this pivotal period in the history of life, we gain deeper insights into the mechanisms that have shaped the evolution of life on Earth and the remarkable adaptability of living organisms. In summary, the dawn of a DNA-based world represents a significant milestone in the evolution of life on Earth. The transition from RNA to DNA as the primary genetic material provided a more stable and reliable means of storing genetic information, supporting the development of complex genomes and regulatory networks. The evolution of DNA polymerases and protein enzymes increased the efficiency and specificity of biochemical reactions, facilitating the growth and replication of cells. The central dogma of molecular biology, DNA to RNA to protein, began to take shape, laying the foundation for the intricate genetic and metabolic networks that characterize modern organisms. The rise of DNA also enabled the development of specialized cellular structures and functions, supporting the evolution of multicellular life and the diversification of ecological niches. The increased genetic complexity allowed for the differentiation of cell types and the formation of tissues and organs, providing numerous advantages in terms of size, specialization, and adaptability. The cooperative interactions between cells within a multicellular organism required sophisticated communication and coordination, facilitated by the intricate genetic and regulatory networks that evolved during this period. As we delve deeper into the evolution of life, the story of the transition from RNA to DNA offers valuable insights into the mechanisms that have shaped the development of biological complexity and diversity. By understanding the molecular innovations that drove this transition, we gain a deeper appreciation for the dynamic and adaptive nature of living systems and the remarkable ingenuity of life on Earth. The rise of DNA as the primary genetic material marked a significant evolutionary advancement, fundamentally transforming the molecular landscape of early life. DNA's double-helix structure, its replication mechanisms, and its inherent stability provided the foundation for the complex genetic and regulatory networks that characterize modern organisms. Understanding the detailed structure of DNA, how it compares to RNA, and the importance of its stability is crucial to appreciating its role in the evolution of life. The structure of DNA is often described as a double helix. a term first introduced by James Watson and Francis Crick when they elucidated its configuration in 1953. This elegant structure consists of two long strands of nucleotides wound around each other, resembling a twisted ladder. Each nucleotide in DNA is composed of three components: a phosphate group, a deoxyribose sugar, and one of four nitrogenous bases: adenine, thymine, cytosine, or guanine. These nucleotides are linked together by covalent bonds between the phosphate of one nucleotide and the sugar of the next, forming a sugar-phosphate backbone that gives the molecule its structural integrity. The two strands of DNA run in opposite directions, a feature referred to as anti-parallel orientation. One strand runs in a 5' to 3' direction, while the complementary strand runs 3' to 5'. This anti-parallel arrangement is crucial for the base pairing that holds the two strands together. The nitrogenous bases on each strand face inward and form specific hydrogen bonds with bases on the opposite strand. Adenine pairs with thymine through 2 hydrogen bonds, and cytosine pairs with guanine through 3 hydrogen bonds. This base pairing mechanism, known as complementary base pairing, ensures that the genetic information can be accurately copied and transmitted. The double helix structure of DNA is stabilized by several forces. Hydrogen bonds between complementary bases provide the primary means of holding the two strands together. Although each individual hydrogen bond is relatively weak, the cumulative effect of thousands or millions of these bonds creates a strong and stable structure. Additionally, the hydrophobic interactions between the stacked base pairs contribute to the stability of the double helix. The nitrogenous bases are hydrophobic and tend to avoid contact with water, leading them to stack tightly on top of one another within the helix. This stacking interaction, often referred to as base stacking, is a significant factor in maintaining the helical structure. The structural stability of DNA is one of its most important characteristics, particularly when compared to RNA. RNA, or ribonucleic acid, differs from DNA in several key aspects. RNA is typically single-stranded, though it can form secondary structures such as hairpins and loops through intramolecular base pairing. The sugar in RNA is ribose, which has a hydroxyl group attached to the 2' carbon, making RNA more chemically reactive and less stable than DNA. RNA contains the base uracil instead of thymine. The presence of the hydroxyl group on the ribose sugar makes RNA more prone to hydrolysis, particularly under alkaline conditions, further contributing to its relative instability compared to DNA. The double-stranded nature of DNA provides a number of advantages over the single-stranded RNA. The complementary strands of DNA enable it to form a stable double helix, protecting the genetic information from environmental damage and enzymatic degradation. The redundancy of having two complementary strands means that if one strand is damaged, the other can serve as a template for accurate repair. This redundancy is critical for maintaining the integrity of genetic information over long periods and through numerous cellular generations. The stability of DNA is also enhanced by the presence of thymine instead of uracil. Thymine is more chemically stable than uracil and is less likely to engage in spontaneous deamination, a common form of mutation. In RNA, cytosine can deaminate to form uracil, which can lead to base pairing errors during replication. The use of thymine in DNA reduces the likelihood of such mutations, thereby enhancing the fidelity of genetic replication and transmission. The replication of DNA is a highly regulated and accurate process, ensuring that genetic information is faithfully passed from one generation to the next. The process of DNA replication begins at specific locations on the DNA molecule known as origins of replication. In these regions, the double helix is unwound by enzymes called helicases, creating a replication fork where the two strands are separated. Single-strand binding proteins stabilize the unwound strands, preventing them from re-annealing or forming secondary structures. Once the DNA strands are separated, each serves as a template for the synthesis of a new complementary strand. This process is catalyzed by DNA polymerases, which add nucleotides to the growing DNA strand in a 5' to 3' direction. DNA polymerases require a short primer, a piece of RNA synthesized by the enzyme primase, to initiate DNA synthesis. The leading strand, which is synthesized continuously, is replicated in the same direction as the replication fork's movement. In contrast, the lagging strand is synthesized discontinuously in short fragments known as Okazaki fragments, which are later joined together by the enzyme DNA ligase. DNA replication is a highly accurate process, thanks to the proofreading ability of DNA polymerases. These enzymes have an intrinsic 3' to 5' exonuclease activity that allows them to remove incorrectly paired nucleotides and replace them with the correct ones. This proofreading function significantly reduces the error rate during DNA replication, ensuring the high fidelity of genetic information transmission. The stability and accuracy of DNA replication are further enhanced by various DNA repair mechanisms. These include direct repair, base excision repair, nucleotide excision repair, and mismatch repair. Each of these pathways addresses specific types of DNA damage, correcting errors that occur during replication or as a result of environmental factors such as UV radiation and chemical mutagens. The ability to repair DNA damage is crucial for maintaining genetic stability and preventing mutations that could lead to diseases such as cancer. The evolution of DNA as the primary genetic material provided a stable and efficient means of storing and transmitting genetic information, facilitating the development of more complex and organized forms of life. The double-helix structure, with its complementary base pairing and anti-parallel strands, ensured the accurate replication and repair of genetic material. The stability of DNA, enhanced by its chemical composition and the presence of thymine, reduced the likelihood of mutations and allowed for the long-term preservation of genetic information. As the primary genetic material, DNA enabled the evolution of larger and more complex genomes. This increased genetic complexity supported the development of sophisticated regulatory networks and metabolic pathways, driving the diversification of life on Earth. The ability to store vast amounts of genetic information in a stable and accurate manner allowed organisms to adapt to changing environments and evolve new traits, leading to the rich biodiversity we observe today. The rise of DNA also had profound implications for the evolution of cellular structures and functions. The increased stability and capacity for information storage provided by DNA enabled the differentiation of cellular functions and the development of specialized compartments within cells. The evolution of eukaryotic cells, characterized by their extensive internal compartmentalization and membrane-bound organelles, was closely tied to the rise of DNA. The nucleus, which houses the cell's genetic material, provided an additional layer of protection and regulation, ensuring the integrity and proper expression of the genome. The increased genetic complexity allowed for the evolution of more sophisticated regulatory mechanisms. Genes could be turned on or off in response to environmental cues, allowing organisms to adapt to changing conditions. Regulatory proteins, such as transcription factors, bound to specific DNA sequences and controlled the transcription of genes, ensuring that proteins were produced only when needed. This level of control and coordination was essential for the efficient functioning of the cell's metabolic and replicative processes. The rise of DNA also facilitated the evolution of multicellular life. The ability to store and regulate vast amounts of genetic information allowed for the differentiation of cell types and the formation of tissues and organs. Multicellularity provided numerous advantages, including increased size, specialization of functions, and the ability to occupy diverse ecological niches. The cooperative interactions between cells within a multicellular organism required sophisticated communication and coordination, facilitated by the intricate genetic and regulatory networks that evolved during this period. In conclusion, the rise of DNA as the primary genetic material represents a significant milestone in the evolution of life on Earth. The double-helix structure of DNA, with its complementary base pairing and anti-parallel strands, provided a stable and efficient means of storing and transmitting genetic information. The increased stability and accuracy of DNA replication and repair facilitated the evolution of larger and more complex genomes, supporting the development of sophisticated regulatory networks and metabolic pathways. The rise of DNA enabled the evolution of eukaryotic cells and multicellular life, driving the diversification and complexity of life on our planet. Through the study of DNA structure, replication, and stability, we gain a deeper appreciation for the remarkable adaptability and ingenuity of living systems, providing a foundation for understanding the continued evolution of life on Earth. The replication of DNA is a fundamental process that ensures the accurate transmission of genetic information from one generation to the next. This complex mechanism involves a series of finely tuned steps orchestrated by a variety of enzymes and other proteins to duplicate the entire genome of an organism. Understanding the intricate details of DNA replication, the roles of key enzymes such as DNA polymerase, and the mechanisms that ensure high fidelity and error correction, provides deep insights into the molecular foundation of life. DNA replication begins at specific sites within the genome called origins of replication. These regions are typically rich in adenine-thymine pairs because the AT bond, involving only 2 hydrogen bonds, is easier to separate compared to the guanine-cytosine bond, which has 3 hydrogen bonds. In prokaryotic cells, such as bacteria, there is usually a single origin of replication on the circular chromosome. In contrast, eukaryotic cells, with their larger and more complex linear chromosomes, have multiple origins of replication to ensure the entire genome is replicated in a timely manner. The initiation of DNA replication involves several key steps. First, initiator proteins bind to the origin of replication, causing the DNA to unwind and form a replication bubble. Helicase enzymes then further unwind the DNA double helix by breaking the hydrogen bonds between the 2 strands, creating 2 single-stranded DNA templates. This unwinding generates torsional strain ahead of the replication fork, which is relieved by topoisomerase. These enzymes cut the DNA, allow it to unwind, and then reseal the break, preventing the DNA from becoming supercoiled and ensuring smooth progression of the replication fork. Single-strand binding proteins stabilize the unwound single-stranded DNA, preventing it from re-annealing or forming secondary structures that could impede the replication process. These proteins bind tightly to the single-stranded DNA, protecting it from nucleuses and maintaining it in a state suitable for replication. Once the DNA is unwound and stabilized, the synthesis of new DNA strands begins. This process is catalyzed by DNA polymerases, a family of enzymes responsible for adding nucleotides to the growing DNA chain. DNA polymerases can only add nucleotides to an existing strand of DNA or RNA, so an RNA primer is needed to initiate synthesis. The enzyme primase synthesizes a short RNA primer complementary to the DNA template, providing a free 3' hydroxyl group for DNA polymerase to extend. DNA polymerases synthesize new DNA strands by adding nucleotides complementary to the template strand in a 5' to 3' direction. As each nucleotide is added, a phosphodiester bond is formed between the 3' hydroxyl group of the growing DNA strand and the 5' phosphate group of the incoming nucleotide. This reaction releases a molecule of pyrophosphate, which is subsequently hydrolyzed, driving the reaction forward and ensuring the addition of nucleotides. In the replication process, the two strands of DNA are replicated differently due to their anti-parallel nature. The leading strand is synthesized continuously in the direction of the replication fork. As the replication fork progresses, DNA polymerase can continuously add nucleotides to the growing strand without interruption. In contrast, the lagging strand is synthesized discontinuously in short segments called Okazaki fragments. These fragments are synthesized in the opposite direction to the replication fork movement, requiring multiple RNA primers. DNA polymerase extends each primer, synthesizing short segments of DNA which are later joined together by DNA ligase to form a continuous strand. The synthesis of the lagging strand involves additional coordination to ensure that each Okazaki fragment is properly initiated and completed. Primase periodically synthesizes new RNA primers along the lagging strand template, and DNA polymerase extends these primers to form Okazaki fragments. As each fragment is completed, the RNA primers are removed and replaced with DNA. This process is facilitated by RNase H, which degrades the RNA primers, and DNA polymerase, which fills in the resulting gaps with DNA. Finally, DNA ligase seals the nicks between Okazaki fragments by forming phosphodiester bonds, creating a continuous DNA strand. That accuracy of DNA replication is paramount for maintaining genetic stability and preventing mutations. DNA polymerases possess an intrinsic proofreading activity that significantly enhances the fidelity of replication. This proofreading function involves a 3' to 5' exonuclease activity, which allows the enzyme to remove incorrectly incorporated nucleotides. When an incorrect nucleotide is added, the polymerase pauses, and the exonuclease activity excises the mismatched nucleotide. The polymerase then resumes synthesis, incorporating the correct nucleotide. This proofreading mechanism reduces the error rate of DNA replication by several orders of magnitude, ensuring high fidelity in the transmission of genetic information. In addition to the intrinsic proofreading activity of DNA polymerases, several DNA repair pathways further enhance the accuracy of DNA replication. Mismatch repair, or MMR, is one such pathway that corrects errors missed by the proofreading activity. The MMR system recognizes and repairs mismatched base pairs that escape the initial proofreading. The repair process involves several steps: recognition of the mismatch, excision of the incorrect segment, resynthesis using the correct template strand, and ligation to restore the integrity of the DNA molecule. Key proteins involved in MMR include MUT-S, which recognizes and binds to the mismatch, and MUT-L, which coordinates the repair process. The stability and accuracy of DNA replication are also supported by other DNA repair mechanisms that address various types of DNA damage. Base excision repair, or BER, corrects small non-helix distorting base lesions caused by oxidation, alkylation, or deamination. The BER pathway involves the recognition and removal of the damaged base by a DNA glycosylase, followed by cleavage of the DNA backbone by an AP endonuclease. DNA polymerase then fills in the gap with the correct nucleotide, and DNA ligase seals the nick. Nucleotide excision repair, or NER, addresses bulky, helix-distorting lesions such as those caused by UV radiation. The NER pathway involves the recognition of the lesion, excision of a short single-stranded DNA segment containing the damage, and resynthesis of the excised region using the undamaged strand as a template. Key proteins involved in NER include the XPC complex, which recognizes the damage, and the XPB and XPD helicases, which unwind the DNA to allow for excision and repair. Direct repair mechanisms also contribute to the stability of DNA by reversing specific types of damage without the need for excision and resynthesis. For example, photolyase enzymes can directly reverse UV-induced thymine dimers by using energy from visible light to break the covalent bonds between the thymine bases. Alkyltransferase enzymes can remove alkyl groups from damaged bases, restoring them to their original state. The combined action of these repair pathways ensures that DNA replication proceeds with high fidelity, maintaining the integrity of genetic information and preventing mutations that could lead to diseases such as cancer. The ability of cells to accurately replicate and repair their DNA is a testament to the intricate molecular machinery that has evolved to safeguard the genome. The regulation of DNA replication is another critical aspect of maintaining genomic stability. In eukaryotic cells, replication is tightly coordinated with the cell cycle to ensure that the genome is replicated once and only once per cell division. The initiation of DNA replication is controlled by a complex network of signaling pathways that respond to internal and external cues. Key regulatory proteins such as cyclins and cyclin-dependent kinases or CDKs modulate the activity of replication initiator proteins and ensure that replication origins are activated at the appropriate time. During the G1 phase of the cell cycle, replication origins are licensed for replication by the assembly of the pre-replicative complex, or pre-RC. This complex includes the origin recognition complex, or ORC, which binds to replication origins, and the mini-chromosome maintenance, or MCM, helicase, which is loaded onto the DNA to prepare for unwinding. The transition from G1 to S phase, where DNA replication occurs, is regulated by the activation of CDKs, which phosphorylate components of the pre-RC and recruit additional factors necessary for the initiation of replication. The tight regulation of DNA replication ensures that each segment of the genome is replicated once per cell cycle, preventing re-replication and maintaining genomic integrity. The coordination of replication with other cellular processes, such as transcription and DNA repair, further ensures the proper maintenance and expression of genetic information. In summary, DNA replication is a highly regulated and accurate process that ensures the faithful transmission of genetic information. The mechanisms of DNA replication involve a series of coordinated steps, including the unwinding of the DNA helix, stabilization of single-stranded templates, synthesis of new DNA strands by DNA polymerases, and the joining of Okazaki fragments. The intrinsic proofreading activity of DNA polymerases and various DNA repair pathways enhance the fidelity of replication, preventing mutations and maintaining genomic stability. The regulation of DNA replication is tightly coordinated with the cell cycle to ensure that the genome is replicated once per cell division, safeguarding the integrity of genetic information. The evolution of DNA as the primary genetic material provided a stable and efficient means of storing and transmitting genetic information, facilitating the development of complex genetic and regulatory networks. The double-helix structure, with its complementary base pairing and anti-parallel strands, ensured the accurate replication and repair of genetic material. The stability of DNA, enhanced by its chemical composition and the presence of thymine, reduced the likelihood of mutations and allowed for the long-term preservation of genetic information. The rise of DNA enabled the evolution of larger and more complex genomes, supporting the development of sophisticated regulatory networks and metabolic pathways. The ability to store vast amounts of genetic information in a stable and accurate manner allowed organisms to adapt to changing environments and evolve new traits, leading to the rich biodiversity we observe today. As we continue to explore the evolution of life, the study of DNA replication and stability provides valuable insights into the molecular mechanisms that have shaped the development of biological complexity and diversity. Through this exploration, we gain a deeper appreciation for the remarkable adaptability and ingenuity of living systems, providing a foundation for understanding the continued evolution of life on Earth. The stability of DNA is a cornerstone of its role as the primary genetic material in living organisms. Its chemical robustness ensures the long-term preservation and accurate transmission of genetic information, providing significant evolutionary advantages over RNA. This section delves into the chemical stability of DNA, its impact on the preservation and transmission of genetic information, and the evolutionary benefits conferred by this stability. DNA and RNA differ significantly in their chemical composition and structural features, which directly influence their stability. DNA, or deoxyribonucleic acid, is composed of a deoxyribose sugar, a phosphate group, and 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. The deoxyribose sugar lacks a hydroxyl group at the 2' position, which is present in the ribose sugar of RNA or ribonucleic acid. This absence of the 2' hydroxyl group in DNA is a crucial factor contributing to its chemical stability. The hydroxyl group in RNA makes it more susceptible to hydrolytic cleavage, especially under alkaline conditions, leading to the breakdown of the RNA strand. In contrast, the deoxyribose sugar in DNA makes the molecule more resistant to hydrolysis, enhancing its stability. Another key difference between DNA and RNA is the presence of thymine in DNA and uracil in RNA. Thymine, which has a methyl group at the 5' position, is more chemically stable than uracil, which lacks this methyl group. The presence of thymine in DNA reduces the likelihood of spontaneous deamination, a common form of chemical damage where cytosine is converted to uracil. In RNA, such deamination events can lead to base pairing errors during replication and transcription, increasing the mutation rate. By using thymine instead of uracil, DNA enhances the fidelity of genetic information storage and transmission. The double-stranded structure of DNA also contributes significantly to its stability. The complementary base pairing between adenine and thymine, and between cytosine and guanine, forms hydrogen bonds that hold the 2 strands together. The stacking interactions between adjacent base pairs further stabilize the double helix. These interactions create a highly stable and protective environment for the genetic information encoded within the DNA sequence. The double helix not only protects the bases from chemical damage, but also provides a mechanism for repair. If one strand is damaged, the undamaged complementary strand can serve as a template for accurate repair, ensuring the preservation of genetic information. In contrast, RNA is typically single-stranded and lacks the structural stability provided by the double helix. While RNA can form secondary structures such as hairpins and loops through intramolecular base pairing, these structures are generally less stable than the double-stranded DNA helix. The single-stranded nature of RNA makes it more prone to degradation by nucleases and other environmental factors. This inherent instability limits the use of RNA as a long-term storage molecule for genetic information, favoring the more stable DNA for this role. The chemical stability of DNA has profound implications for the preservation and transmission of genetic information. The ability of DNA to resist chemical damage and maintain its integrity over long periods is essential for the accurate replication and inheritance of genetic traits. The stability of DNA ensures that the genetic information encoded within it can be faithfully copied during cell division, allowing for the propagation of genetic material across generations. This high fidelity in replication is crucial for maintaining the continuity of genetic information and preventing the accumulation of deleterious mutations. The stability of DNA also facilitates the evolution of larger and more complex genomes. In early life forms, the stability provided by DNA allowed for the accumulation of more genetic material, supporting the development of new genes and regulatory elements. This increase in genetic complexity enabled the evolution of sophisticated metabolic pathways, regulatory networks, and cellular structures. The stability of DNA provided a secure foundation upon which the diversity and complexity of life could build, driving the evolutionary processes that led to the rich biodiversity we observe today. One of the key evolutionary advantages conferred by DNA stability is the ability to support complex and multicellular life forms. The stability of DNA allows for the precise regulation of gene expression, which is essential for the development and differentiation of various cell types within a multicellular organism. The accurate transmission of genetic information ensures that each cell in the organism receives the correct instructions for its function, enabling the coordinated development and maintenance of tissues and organs. This level of complexity and organization would be difficult to achieve with the less stable genetic material like RNA. The stability of DNA also enhances the ability of organisms to adapt to environmental changes. The reliable preservation of genetic information allows for the accumulation of beneficial mutations over time, driving the process of natural selection. The ability to maintain genetic stability while allowing for evolutionary change is a delicate balance that DNA achieves through its structural properties and repair mechanisms. This balance ensures that genetic information can be preserved and accurately transmitted while also providing the raw material for evolution. As we continue to explore the evolution of life, the study of DNA stability provides valuable insights into the molecular mechanisms that have shaped the development of biological complexity and diversity. Through this exploration, we gain a deeper appreciation for the remarkable adaptability and ingenuity of living systems, providing a foundation for understanding the continued evolution of life on Earth. The stability of DNA is not just a molecular characteristic. It is a fundamental property that has driven the evolutionary processes that have led to the rich tapestry of life that we see today.